everyone, welcome to Biotech Learning. This is our third video and today we are going to talk about receptors. Our first video told you what is cell signaling and what's its importance. Our second video told you about the ligands or the chemical molecules involved in cell signaling. So today we shall talk about receptors. Receptors are basically proteins that receive these information and transduce it further. So there may be numerous receptors in most of the cells. So today let's see how does a receptor interact with a ligand. It's just like the lock and key model. It's just that the keys only of a specific shape can fit into the locks. In the same way, only ligands of a particular structure can fit into the receptor. So it's basically like the lock and key hypothesis. So after the binding of the ligand with the receptor, there's a conformational change that can lead to further transmission of the signal. Here the example I've taken is that of an insulin receptor. And this is the ligand. When the ligand binds to the receptor, there's a conformational change. So this can lead to the activation of some reaction. It's not always an activation. Sometimes it can also lead to inhibition of some reaction or some sequence of events. Sometimes a pore opens up, a channel opens up within the protein, within the receptor, and it allows some ions like sodium, potassium, calcium to move into the cell or outside the cell. Sometimes the receptor may have some intrinsic enzymic activity and it will catalyze some reactions. Sometimes the receptor may itself not have any enzyme or transporter associated with it, but it will be linked with some enzyme or transporter or some protein which have some enzymic activity. Sometimes the receptor may be itself a DNA binding protein. So when the ligand binds to the receptor, there's a conformational change and the DNA binding properties of this receptor changes. Sometimes the cellular localization properties changes. And as we have talked about the enzymic activity, that can lead to a sequence of events, and that is called the signal transduction cascade, which we'll deal in detail. So let's see the various responses. When a ligand binds to the receptor, it can show some enzymic activity. Sometimes it can show some ion channel opening or closing activity. Sometimes the receptor may be not directly having some enzymic activity, but it will be linked with a protein which has some enzymic activity, or it may be linked with other transporters. Sometimes the receptor may be a DNA binding protein and its DNA binding property changes and cellular localization property changes. And this enzymic activity can also lead to a signal transduction cascade. So all these are the various changes that can happen when the ligand binds to the receptor. So this is just like the relaying of a signal. That is, the signal which is coming from outside is received by the receptor, and the signal or the information is passed onwards or forwards. So the information is passed on from molecule to molecule sometimes. So it's just like the relaying of a signal. So this can be considered as the action of a receptor. But there is also another action called amplification. That is, the effect of a single ligand can be made multiple folds, can be increased by multiple folds. This is called amplification. This could be due to the cooperative interaction between receptors. So this is another property of receptors. It can show amplification. And another important property is integration. That is, when the signal or the ligand binds to the receptor and it will cause a sequence of events. This sequence of events or the biochemical pathway is not isolated. It's part of a bigger network. So the binding of a single ligand to a receptor does not just cause one response. It is a part of a network, and the molecule in this pathway can be linked to molecule of other pathways, and there will be a coordinated response. So this is called integration. So these are the various actions of receptors: relaying of the signal, that is sending the signal onwards, amplification, increasing the effect of a single ligand, or integration, allowing the signal to be incorporated into another biochemical pathway. So let's have a quick recap and then move forward. So we have seen the receptor-ligand interaction. It's just like the lock and key model. We have seen the various responses, and we have also seen the actions of receptors, that is relaying, amplification, and integration. So with these basic information, let's move on to see the types of receptors. So there are basically two types of receptors: extracellular receptors and intracellular receptors. So in this video, we'll be able to cover, or we'll be able to give you a quick overview of only extracellular receptors, and intracellular receptors we'll discuss in the next video. So extracellular receptors, as the name indicates, these are receptors located on the surface of the cell, that's in the plasma membrane. So these are also called cell surface receptors, or transmembrane receptors, or cell-specific proteins or markers. Let's see in detail. So how are these receptors distributed? Now, each cell membrane can have several kinds of receptors with varying surface distribution. Even a single receptor may also be differently distributed at different membrane positions depending on the sort of membrane and cellular function. Basically, receptors are often clustered on the membrane surface and not evenly or uniformly distributed. So the thing we have to keep in mind is that each and every receptor can be differently distributed, and these can be often clustered. It's not uniformly distributed. So now let's see the chemical structure of these receptors. These are basically specialized integral membrane proteins. So what are integral membrane proteins? Let's look at this picture. This shows a lipid bilayer, or the plasma membrane. And here you can see two layers of lipids. So this is why it's called lipid bilayer, and you can see many proteins studded into this. So there are mainly two types of proteins. The first one is the peripheral proteins. These are temporarily associated. That means these can be easily removed. By high pH or high salt concentration, these are loosely attached to the plasma membrane. But the second type, and the topic of our concern, is this integral membrane proteins. These are permanently attached to the plasma membrane. It's very difficult to remove them. You may need detergent action or something like that to remove these proteins. Okay, see, this one just looks like the peripheral protein, but the thing is that these are tightly attached. So it is integral membrane protein by covalent interactions, etc. So integral membrane proteins can be of, again, two types. The one just facing one side of the plasma membrane of the cell, that's why it's called monotopic membrane protein. Mono means one. So these are just facing one side of the plasma membrane. And these are bitopic membrane proteins because they can be seen on both sides. So they are transversing the plasma membrane. So these are called transmembrane proteins. So two basic types of integral membrane proteins are monotopic membrane proteins and this type. That is, they are called polytopic membrane proteins. They are not called only bitopic because there are also some proteins which traverse the plasma membrane many times. So the two types of integral membrane proteins are monotopic membrane proteins and polytopic membrane proteins. And extracellular receptors are mainly of this type, polytopic membrane proteins or transmembrane proteins. So once again, let's see the two types, monotopic membrane proteins and polytopic membrane proteins or transmembrane proteins. And extracellular receptors are basically of this type. So these are called polytopic because there are many proteins which traverse the plasma membrane many times. And there are bitopic membrane proteins also come in this category. So basically, the fact you have to take from this is that extracellular receptors are transmembrane proteins or polytopic membrane proteins. It can be of this type or it can be of this type or this type. Now let's see what are the ligands for extracellular membrane proteins. Since these are extracellular receptors, they receive the signal from outside the cell. So the signals could be made from another cell which is synthesizing these signals, and these signals could be transported via bloodstream and they reach the target cell. So ligands, so the molecules that are the ligands for these type of receptors are hormones, neurotransmitters, cytokines, growth factors, cell adhesion molecules, or nutrients. So they will interact with the receptor to induce changes in the metabolism and activity of the cell. So we have seen the distribution of extracellular receptors, the chemical structure of the receptor, and the ligands. So with this basic information, let's move on to the type or the classes of extracellular receptors. Extracellular receptors are basically classified into three classes. The first is G-protein-coupled receptors. And the second is enzyme-linked receptors. And the third is ionotropic receptors. So I'll give a quick overview of these three classes in this video. The first one is G-protein coupled receptors. They are also called GPCRs. They are the largest family of cell surface receptors, the major class. They are found only in eukaryotes. That's another important point to be noted. They are found only in eukaryotes, animals, and even yeast. Now let's move on to the structure of GPCRs. They are composed of 7 transmembrane alpha helices. This blue color part is the receptor. And this brown circle here you can see is the ligand. And this pink color portion is the G protein that is associating with this receptor. That's why this receptor is called G protein coupled receptors. And when you look at this receptor has 7 transmembrane alpha helices. So they are also called sometimes serpentine receptors or 7TM receptors. And here the loops connecting these alpha helices, they are found in the extracellular side and in the intracellular side. These form the extracellular domain and intracellular domain. And this receptor is associated with a G protein, and this G protein has 3 subunits, alpha, beta, and gamma. And this protein is called G protein because it's associated with guanine nucleotide. Here it is GDP. So this is the structure of a G protein coupled receptor. Now let's see the ligands for this type of receptor, the major class of extracellular receptor. So there are a variety of extracellular signals that can bind to these type of receptors. And they help regulate incredible range of bodily functions from sensation to growth to hormone responses. So the various ligands are photosensitive compounds, light sensitive compounds, odors, pheromones, hormones, and neurotransmitters. And neurotransmitters especially that acts slowly, the slow neurotransmitters like dopamine. So now let's see how does the ligand bind to the receptor and how does the change or the sequence of events that happens. So in this first picture you can see there is a ligand here. The second picture, the ligand binds to the receptor. And now you can see there's a conformational change here. This conformational change leads to the activation of this alpha subunit of this G protein. The activated alpha subunit exchanges this GDP for a GTP. When the GTP binds to the alpha subunit, it dissociates from this beta-gamma dimer. And now the activated alpha subunit can further affect intracellular signaling proteins or target functional proteins directly. So that there are 2 principal signal transduction pathways involved here, that is cAMP signaling pathway and phosphatidylinositol signaling pathway. So we'll be learning about all these signaling pathways in detail in the coming videos. And for now, let's move on to the second type, enzyme-linked receptors. So these are the second major type of extracellular receptors, and the point to be noted is that they are found in all living species. Okay, so now let's move on to see the structure of this receptor. As you can see here, it is a single polypeptide, a transmembrane protein, but it has an integral enzymic domain. So it's also sometimes called catalytic receptor. And in the external side, it has the signaling molecule binding domain. So the enzyme here can be kinases, phosphatases, etc. I think we should know what are kinases and phosphatases. So kinases are enzymes that add a phosphate group to the target protein. Just look at this target B. So to the substrate, it adds a phosphate group, this kinase enzyme. They take phosphate from high energy molecules like ATP. Here ADP and P is ATP. So this phosphate comes and attaches here, and then ADP is released. So kinase does phosphorylation, adding of phosphate to the target protein. So that's the action of kinases. And phosphatases are another group of enzymes that works just the opposite. It removes phosphate from this molecule. It removes phosphate from this target protein. And then when the phosphate is removed, it's called dephosphorylation. The phosphate is removed and it becomes target A. So enzyme-linked receptors have these kinases and phosphatases as a part of their integral enzymic domain. So this enables binding and activation of many intracellular signaling proteins, and this leads to changes in gene transcription and many cellular functions. So let's see the various types or various families, 5 major families of enzyme-linked receptors. So these are the 5 major families, and here you can see these are receptor tyrosine kinases, tyrosine kinase associated receptors, receptor-like tyrosine phosphatases, receptor serine threonine kinases, receptor guanylyl cyclases, and this histidine kinase associated receptor is also analogous to the first one. So you can see there is kinases, phosphatases. So that's why we have talked about kinases and phosphatases. Now what is guanylyl cyclases? These are enzymes that convert GTP to cyclic GMP and pyrophosphate. And histidine kinase associated receptors are mainly found in bacteria and fungi. So here you can see many families have kinases in them. Then why are they classified separately? This is because kinases, as we have said, adds a phosphate group to the target protein. But if the phosphate group is added to specifically a tyrosine moiety of this target protein, then it is called tyrosine kinase. So receptor tyrosine kinases are enzyme-linked receptors that have an integral enzyme, and that enzyme is a kinase that adds phosphate to specifically tyrosine moiety of the target protein. So basically these are receptors, enzyme-linked receptors that have an intrinsic tyrosine kinase enzyme. I hope that made it a little more clear. And what is receptor serine threonine kinases? These are enzyme-linked receptors that have an intrinsic serine threonine kinase. What about this? These are receptors that have tyrosine phosphatase enzyme in them. What about this one? These are a special type. These are enzyme-linked receptors that do not have an intrinsic enzymic domain. So they associate with other proteins that have an enzymic domain, especially this tyrosine kinase activity. So these are enzyme-linked receptors that associate with other proteins that have tyrosine kinase domain. So that's why they are in general called enzyme-linked receptors. So we cannot discuss all these in detail in this video, but we shall discuss a little bit about the first one, receptor tyrosine kinase. So as you can see here, this is a single polypeptide. On the extracellular side, it has a hormone binding site or a ligand binding site, and on the intracellular side, it has this enzymic domain, and that is specifically the tyrosine kinase domain. Majority of growth factor receptors consist of receptor tyrosine kinase, epidermal growth factor, platelet-derived growth factor, fibroblast growth factor, hepatocyte growth factor, nerve growth factor, and hormones such as insulin. All these growth factor receptors belong to this class. So let's see, how does this happen? Here you can see two receptor tyrosine kinase, and when the peptide hormone binds here, the first step is that the receptors come closer and they dimerize. Now we have talked a lot about this enzyme phosphorylating a target protein. Now who is the target for this enzyme? The target is the opposite enzyme, and the target for this enzymic domain is the opposite enzymic domain. So they phosphorylate each other, they autophosphorylate, and this leads to catalyzing further intracellular reactions. So this is how it works. And now let's move on to ionotropic receptors. Ionotropic receptors are a group of transmembrane ion channel proteins. That is, they have a channel through which ions can move inside or outside the cell. So it can be sodium, potassium, calcium, chlorine, etc. Now the point to be noted is that it has a separate site for the binding of the ligand or the signaling molecule, and there is a channel for the movement of ions. These ions are not the ligands. The ligand binding site is different. When the ligand binds to this protein, which is a transmembrane protein, a channel opens up and extracellular ions can move inside. And the ligand binding here can be some neurotransmitter, especially fast neurotransmitters like acetylcholine and GABA. Let's see this picture. Here you can see acetylcholine is the ligand here. When this binds to this receptor, a channel opens up and then sodium ions move towards the inside of the cell. It's as simple as that. These are ionotropic receptors. So I think I gave you a quick overview about the three types of extracellular receptors and some basic information. In the next video, we shall talk about intracellular receptors. Hope this video was clear to you all. Please put your doubts in the comment section below and enjoy learning.